Module 2, Advanced Proteomics and Recombinant DNA Technology. Section 1, Introduction to Recombinant DNA Technology. Recombinant DNA technology, often referred to as genetic engineering, involves combining DNA molecules from different sources to create a new genetic sequence that does not exist naturally. This powerful technique has revolutionized the field of molecular biology, medicine, agriculture, and biotechnology. The process begins with identifying the gene of interest and isolating it from the source organism. This gene is then inserted into a vector, most commonly a plasmid, which is capable of independent replication. The vector carrying the recombinant DNA is introduced into a host organism, usually a bacterium such as Escherichia coli, where it is replicated and expressed. Recombinant DNA technology offers a way to produce large amounts of proteins, such as insulin or growth hormones. Create genetically modified crops with resistance to pest or environmental stress. Study gene function and regulation. Develop vaccines and novel therapeutics. Section 2, Gene recombination mechanisms. Gene recombination is the process through which genetic material is rearranged to create new combinations of DNA sequences. In recombinant DNA technology, this principle is applied artificially, but it also occurs naturally through several mechanisms. Bacterial conjugation. This is the direct transfer of DNA between bacterial cells through physical contact, usually via a structure called the pilus. One bacterium acts as the donor, transferring plasmid DNA to the recipient. Conjugation allows rapid spread of traits such as antibiotic resistance. Transformation. Transformation occurs when bacteria take up free DNA fragments from their environment. The DNA can integrate into the host genome, leading to the expression of new traits. In the lab, scientists exploit transformation to introduce recombinant plasmids into bacteria. Transduction. This process involves bacteriophages, viruses that infect bacteria. During viral replication, fragments of bacterial DNA can be mistakenly packaged into new virus particles. When these infect another bacterium, they introduce the DNA from the previous host. Episomes and plasmids. Episomes are plasmids capable of integrating into the host genome. Plasmids are small, circular DNA molecules independent of chromosomal DNA and are widely used as vectors in recombinant DNA experiments due to their ease of manipulation. Section 3, Gene transfer techniques. Recombinant DNA work often requires introducing genetic material into host cells. Various physical, chemical, and biological methods exist for gene transfer. Microinjection. A fine glass micropipette injects DNA directly into the nucleus of a cell. This method is precise but labor-intensive and requires specialized equipment. Electroporation. Cells are exposed to a brief electrical pulse, creating temporary pores in the cell membrane through which DNA can enter. Electroporation is efficient for a wide range of cell types. Microprojectile or ballistic method. Also known as the gene gun method, microscopic particles coated with DNA are physically shot into cells at high velocity. This technique is especially useful for plant transformation. Shotgun method. In genomics, the shotgun approach involves randomly breaking DNA into small fragments, cloning them into vectors, and sequencing them to reconstruct the genome. Ultrasonication. High-frequency sound waves disrupt the cell membrane, allowing DNA uptake. While effective, this method can cause significant cell damage. Liposome-mediated transfer. Liposomes, artificial lipid vesicles, encapsulate DNA and fuse with cell membranes to deliver genetic material inside the cell. Microlaser. This technique uses focused laser beams to transiently open cell membranes, facilitating DNA entry. It offers precision in targeting specific cells. Section 4, Introduction to Proteomics. Proteomics is the large-scale study of proteins, their structures, functions, and interactions. Unlike the genome, which is relatively stable, the proteome is dynamic, changing in response to environmental factors, developmental stages, or disease conditions. Proteomics aims to identify proteins expressed in a particular cell or tissue, quantify changes in protein abundance, study protein modifications and interactions, understand protein function in the context of cellular pathways. Section 5, Protein structure and function. Proteins are polymers of amino acids, and their function is determined by their structure. Primary structure, sequence of amino acids. Secondary structure, local folding into alpha helices or beta sheets. Tertiary structure, overall three-dimensional shape of the protein. Quaternary structure, assembly of multiple polypeptide subunits. A protein's function can range from catalyzing biochemical reactions, enzymes, to providing structural support, collagen, transporting molecules, hemoglobin, or transmitting signals, receptors. Section 6, Techniques in Proteomics. Proteomics relies on various analytical methods. Protein fractionation, separation, and purification. Proteins can be separated based on size, charge, hydrophobicity, or affinity for specific ligands. One-dimensional and two-dimensional polyacrylamide gel electrophoresis, 1D page and 2D page. 1D page separates proteins based on molecular weight. 2D page adds separation by isoelectric point, providing high-resolution protein maps. Protein identification. After separation, proteins can be identified using mass spectrometry, immunodetection, or sequencing. Section 7, Mass spectrometry in proteomics. Mass spectrometry, MS, is a core technology for analyzing proteins. It measures the mass-to-charge ratio of ionized particles, allowing precise identification and characterization of proteins. Types of mass spectrometry. MODTOF, matrix-assisted laser desorption/ionization, time of flight. ESIMS, electrospray ionization mass spectrometry. Tandem MS, MS/MS. Applications include determining protein sequences, identifying post-translational modifications, and studying protein complexes. Section 8, Protein databases and bioinformatics. Bioinformatics tools are essential for analyzing proteomics data. Protein databases, such as UniProt and Protein Data Bank, PDB, provide curated information on protein sequences, structures, and functions. Analytical software assists with matching MS data to protein sequences, predicting protein structure, analyzing interaction networks. Section 9, Experimental design and quality control. Careful planning ensures reliable results. Biological replicates to account for variability. Technical replicates to assess reproducibility. Internal standards for quantitation. Quality control measures to avoid contamination or instrument drift. Section 10, Advanced proteomic applications. Protein array technology, high-throughput method to detect protein interactions or biomarkers. Post-translational modification analysis, phosphorylation, glycosylation, and acetylation often regulate protein function. Peptide mass fingerprinting, identifying proteins by matching peptide masses to databases. Fragment fingerprinting, determining amino acid sequence from fragmented peptides. Section 11, Applications in scientific research. Proteomics, combined with recombinant DNA technology, has numerous applications. Identifying disease biomarkers for early diagnosis. Understanding drug mechanisms and resistance. Mapping signaling pathways in cancer or neurodegenerative diseases. Developing personalized medicine approaches. Section 12, Ethical, Safety, and Regulatory Considerations. Recombinant DNA and proteomics research must comply with ethical guidelines, biosafety standards, and legal regulations. This includes risk assessment, containment measures, and ethical review for experiments involving human or animal samples.